we know that life starts from the zygote, and then the zygote divides, forms an embryo. This embryo results in a fetus, right? And from the fetus, you come up with a child. Now the thing is, a embryo থেকে যে fetusটা হইলো, fetus, it has different organs, different tissues, তাই না? For example, you have এটাকে যদি কেটে টুকরা টুকরা করে ফেলো fetusটাকে, say you're performing black magic or something, so you need to sacrifice a fetus. So fetusের nerve tissue বের করলা, muscle tissue বের করলা, cardiac tissue বের করলা বা cardiac cells গুলো বের করলা, white blood cell বের করে আনলা। Now black magic perform করা আমাদের টাস্ক না, আমাদের যে কনসেপ্টটা সেটা হচ্ছে এই একটা সেল থেকে কি করে এতগুলো ভ্যারিয়েশন অ্যারাইজ করলো, বিকজ এইখানে যে জেনেটিক ইনফরমেশন আছে, যে জেনেটিক ম্যাটেরিয়ালস আছে, এক্স্যাক্টলি সেম জেনেটিক ম্যাটেরিয়াল তো এই সব কয়টা সেলের মধ্যেই আছে। যে জেনেটিক ইনফরমেশন এই নিউরনটার মধ্যে আছে, এক্স্যাক্ট সেম জেনেটিক ইনফরমেশন এই হোয়াইট ব্লাড সেলটার মধ্যে আছে। So the question is, if they have the exact same genetic information, why are they so different? Yes, exactly. We talked about gene expression, right? All of them have the same genes. These has, say, 25,000 genes. These also has the exact same 25,000 genes. But the difference is, a 25,000 genes er moddhe neuron ki sob koyta activate korche? No. It has selectively turned on a few genes, say 10,000 genes, while switched off the other genes. This one has selectively turned on certain varieties of those genes and switched off the other genes. Some of the genes that were switched on were common. For example, both have cytoplasm, both have cell membrane. So ওই জিনগুলা সবার জন্যই অন করা আছে। But then this has special neurotransmitter যেটা এইটার মধ্যে নাই, which means neurotransmitter has switched on the genes that codes for the neurotransmitter, while white blood cell has selectively switched off those genes. In contrast, white blood cell দেখা গেছে যে it has turned on the genes for hydrolytic enzymes, but those genes were switched off in this cell. Now, do you remember these things from O levels? Okay. So with that clarified, আজকে we are going to know this gene expression, this concept of gene expression. We are going to dig deeper into this. So the concept of gene expression is nothing new for us, right? আমরা এটা already study করে আসছি. What is new in AS are these three terminologies. We need to understand what they mean, and these terms are, number one, transcription factor, promoter sequence, and enhancer sequence. So what does they mean? Let's start with transcription factor. So in your book you'll find both of these definitions, and it says transcription factors are proteins that bind to DNA in the nucleus and affect the process of transcribing the genetic material. So basically একটা জিন এটা এক্সপ্রেসড হবে কি না সেটা কে ডিটারমাইন করতেছে? This transcription factor. এটা আমি আরেকটু ডিটেইলস আরেকটু পরে বলবো, just stay with me. We have another associated definition, which is promoter sequence. These definitions are also in the book, so let's read them. Promoter sequence are specific region on the DNA to which transcription factors bind to stimulate transcription. Now, both these things will become clear as I show this picture. So this is the best way to understand transcription factor and promoter sequence. Imagine this is the length of DNA, and we know length of DNA comprises of sections of genes. So this is a gene, this is a particular gene. Let's say this is gene 1. Now at the beginning of this gene there is another segment of DNA whose job is not to code for protein, rather it is like a switch on factor for gene 1. এইটাকে আমরা বলছি this is promoter sequence. Every gene has its own promoter sequence. Understood? You know, every book, there is a preface to that book, introduction to that book. So basically this is like the preface of the gene. Okay, now what about the transcription factor? Before going to transcription factor, let's review how genes are actually expressed. We know normally genes are present in this coil form, twisted alpha helix form. Before this can be read, first it has to straighten up, right? Then the hydrogen bond needs to be broken, and we say that the DNA has first unwind, then it has unzipped, unzipping হচ্ছে, right? Now এই কাজগুলা কে করে? You need an enzyme. Which enzyme? RNA polymerase. In order to synthesize protein, you need RNA polymerase. Okay. Okay. So here is the RNA polymerase, and like I have said, in order for this gene to express, the RNA polymerase has to start working on it. But then RNA polymerase এর কাজ করার জন্য it has to sit somewhere. Where is it going to sit? On the transcription factor. And transcription factor, first you have to fit it somewhere. Where is this transcription factor going to fit? In the promoter sequence. So first you have to find out the promoter sequence. The transcription factor goes on top of the promoter sequence. Now it has provided the stage for the action of RNA polymerase. So now RNA polymerase sits on the transcription factor and does it what it does. The gene is synthesized, gene is read, and it produces an mRNA. Any questions? Did you understand the concept of transcription factor and promoter sequence? Clear? Any questions?
Mutation is a promoter gene. Gene 1
Yes.
Promoter gene to recognize gene 1 আসলে.
Okay. So actually this is two different questions. একটা হচ্ছে যদি জিনের মধ্যে mutation হয়, we already know this from molecules. It is going to synthesize an abnormal version of the protein. If there is a mutation in the promoter sequence, that means this platform has been changed. So the chances are a transcription factor এখানে বাইন্ডই করতে পারবে না, which means RNA polymerase আসতে পারবে না, এই জিনটা তখন কি হয়ে যাবে? সাইলেন্ট হয়ে যাবে, unexpressed থাকবে. So this is one possible outcome. Now হইতে পারে, it can occur differently. Something else might also happen, but this is one of the possibilities. সেই promoter sequence mutation হইলে the entire gene might be silenced. So we have studied promoter sequence, we have studied transcription factor. What's left? Enhancer sequence. Enhancer sequence can be expressed by another picture, which is this one. So basically, enhancer sequence is like a signboard or an indicator showing the transcription factor where to start its job. It's defined in the book as such. Enhancer sequence, specific region of DNA to which transcription factor bind and regulate the activity of DNA by changing the structure of the chromatin. In all of us, we also studied about chromatin. We know that DNA inside the cell, they don't exist as a straight line. Rather, they undergo coiling, and thus they form chromatids, right? That we know. এখন কথা হচ্ছে this particular sequence of chromatin where everything is coiled. Do you think this area, if there is a gene over here, do you think at this stage this gene can be read? No, because it is highly coiled, so there is no way that RNA polymerase will be able to fit and read this gene. So in order to activate this target gene, first you have to untangle this section of the chromatin. And this is where enhancer sequence comes to play. Located over here is an enhancer sequence, and for this enhancer sequence there are separate transcription factors. So when this target gene needs to be switched on, a enhancer sequence ki korbe or target transcription factor ke call korbe, it's going to come over here and bind with the enhancer sequence, and this is going to untangle this section of the chromatin. Now the gene is free to be expressed, and we know what happens next. Promoter sequence total ekhane expose hoye jabe, so the RNA polymerase will come here and this gene can be read. There are some enhancer sequence which can do the reverse, that is, it can also hide a particular gene. So it can work both ways. So that's the function of the enhancer sequence. Basically, promoter sequence and enhancer sequence, they have much in common. However, the main difference, if you asked me, the main difference is that promoter sequence are located just in front of the target gene, whereas enhancer sequences are located farther from the target gene. And promoter sequence always start or is always present at the beginning of the gene, whereas enhancer sequence can stay at the beginning or it can stay further away from the gene at the back. Bottom line is that enhancer sequences increases the overall efficiency of the transcription process. Okay? Any questions? Yes. Yes. Yeah, they also have some role. There are many factors which are influencing the coiling and uncoiling. One of those factors is this enhancer sequence. Histone also plays a role. We'll come to that later. Yeah.
Enhancer sequence is একটা gene target করবে অন্য gene
a single enhancer sequence can target or influence expression of multiple genes, whereas promoter sequence are usually one promoter sequence usually helps in the transcription of one single gene. Understood?